Hello, my name is Devin Hall.
And I'm Marley Peck, and this is our experiment on how the effects of motor oil in soil affect plants. So for our project, we decided to experiment with 15 groups of soybean plants in different levels of motor oil contaminated soil. And this was used to simulate motor oil runoff into soils that we grow our crops in. So this runoff can come from cars, boats, lawnmowers, or farm equipment. And this oil can make its way into waterways and also run off sides of slanted grounds and soak into the soil. And this can affect us in many ways. So that is why we decided to use oil as our chosen addition into the plants in our experiment. So through the growth of these soybeans in the lab, we were able to take measurements based on germination percentage in each pot and the height of the plants in each pot. And we controlled this experiment by planting the same amount of soybean seeds in each pot, which was 6, and then we also watered them the same every day. Sometimes we changed the amount of water given, but no matter what, each of the plants was watered the same relative to that day. So we hypothesized that the motor oil would have adverse effects on the plants, but we actually ended up getting very sporadic results throughout the experiment. In some plants, the motor oil seemed as if it would be beneficial to some of the plants, and then other plants showed no difference in germination and height based on the amount of contamination that they were faced with. And so we ended up running these results through a statistical ANOVA, and this will be discussed later. Overall, the results were deemed insignificant, but we may have a reason as to why. According to Avoy and her colleagues, soil and surface water contamination by used oil is a common occurrence in most developing countries, and this has been shown to have harmful effects on the environment and human beings at large. So some of the substances that can contaminate soil are things like road salts, and this can dehydrate the soil, but another common substance is motor oil, and this is what we have chosen to discuss. This is because many of us drive cars, and we could be contributing to this, and also many agricultural fields reside next to busy roads, so this could make them very susceptible to this sort of contamination. And so as I said previously, we wanted to see how motor oil runoff affects the soil and how the plants will grow in our soil. So for our experiment, we created 5 overall levels of motor oil contamination. So the first group was the control group with no oil at all, and then from there the 4 other groups went up in concentration. So they varied from 0 milliliters of motor oil to 6 milliliters of motor oil mixed into the soil. And for 2 weeks, we cared for the plants and collected important data to keep track of germination percentages and also the plant height. And we used numerical statistics because this would help us be able to run the stats through the statistical ANOVA, and this would allow us to see the significance of the results and whether there were differences in plant growth and germination based on how high a concentration the plants resided in. So experiments like this can help scientists and researchers examine the effects of motor oil and how these plants are able to perform phytoremediation within adverse conditions. Phytoremediation is a plant's ability to remove contaminants from the soil, and Devin will be discussing further on that when she discusses the results of the experiment.
Okay, so going more into specifics, so we planted 6 soybean seeds into each pot. Starting with the control, it had 3 replicates with no motor oil, and then the next level had 1.5 milliliters, the level after that 3 milliliters, 4 milliliters, all the way up to, or 4.5 milliliters, and then all the way up to 6 milliliters in each pot with the replicates. Throughout the 2 weeks, we tended to it every day. We wrote down descriptions of the plant, of the weather that day, how much sun it was getting, how much we watered it, which varied throughout the process. We recorded that data every day, and then at the end of each week, we collected germination and height. After the first week, we didn't really have much going on, so that kind of freaked us out for a second because we thought we might have to restart the experiment. Maybe we put too much motor oil, maybe we didn't water it enough. We definitely didn't water it enough, so we watered it more. That fixed it. So you have the tables over here of what the average germination and the average plant height for the last 2 weeks, and then the ANOVA results that said that there was not enough variance to be statistically significant, so it did not support our hypothesis. Most plants in the higher concentrations were dead, like in the 4th level, but weirdly enough, level 2 had the most germination and the best height out of the group. So it made us wonder if maybe there was something in the motor oil that was making it easier to live. And then we found out about phyto remediation, which specifically in a study done by Leo mentions similar results where a soybean would remove up to 34.2% of oil in the soil to make it more livable and more survivable for that plant. So if we wanted to study specifically what oil does to plants, soybeans were probably one of the worst plants to choose for this. Going back, I also don't think we added enough motor oil to the plants. Knowing this information now, we probably would have done more extreme levels of oil throughout the process, but we were worried about, one, totally destroying the experiment with too much motor oil, and two, not putting enough in. Like, we wanted to find that middle ground where you see the different, like the big change, and we did not achieve that. So going back, we would try to achieve that.
Okay, and then going over our results specifically, so pictured right here is the plants on their final day of measurements, and the plants that have 0 milliliters of motor oil are furthest to the left, while the ones with 6 milliliters of motor oil mixed into the soil are furthest to the right. And so as you can see, our results do seem very inconsistent. So as Devin mentioned, the average germination, the highest level was in level 2 with 44.4% germination, and the levels level 4 with 6 milliliters of motor oil was at a close second with 38.9% germination, so that does seem a little weird. For the average plant height, the highest average was 6.17 centimeters in the control group, and this was about the only stat that we had that actually did make sense. And so taking all of these statistics into considerations, we ran them in a statistical ANOVA, and so in the end the p-value calculated for plant height was, and this was compared to for plant height compared to oil contamination was 0.729, and the one for the percentage of seeds germinated was 0.268, and so for these results to be deemed significant, they would have to be less than 0.05, which didn't end up happening, so that's why our results are deemed insignificant, and we came up with a reason why it could be because of phytoremediation.
I would also like to point out that we had a really interesting pot in the 4th level. It's actually like this top right corner where everything's dead. 4 seeds were germinated, which was one of the highest levels of germination we had. They all grew to exactly 2 centimeters and then died. And I don't know what that is, but I thought it was interesting. So thank you.